class. So this week we will continue on chapter 4, but today we will only cover two subtopics, which are DNA replications and also transcription process. So before we try to understand the process of transcription and DNA replication, we need to know about the DNA. So the DNA structure was discovered in 1953 by James Watson and also Francis Crick, in which they discovered that DNA is a double helix structure and DNA is the important genetic material. So DNA or RNA is actually nucleic acids, and nucleic acids are the polymer of nucleotides, meaning to say nucleotide is the monomer for nucleic acids. So nucleotide consists of three components. The first one we have nitrogenous base, second one is the pentose sugar, and also one phosphate group. So the sugar in DNA, the sugar is deoxyribose sugar. Meanwhile, in RNA is ribose sugar. The difference between deoxy and ribose sugar is that the presence of oxygen atom on the carbon number 2 here. And then next is DNA or RNA only consists of one phosphate group. Meanwhile, for the nitrogenous bases are divided into two groups, either pyrimidines or purines. So pyrimidines structure, they only have a single carbon-nitrogen ring here, as you can see. Meanwhile, for purines, the structure composed of two carbon-nitrogen ring here that fuse together. Okay, 5-member ring fused with 6-member ring here. So cytosine and thymine in DNA under the group of pyrimidine. Meanwhile, cytosine and uracil are under the group of pyrimidine in RNA. Meanwhile, both of DNA and RNA, the purine consists of adenine and also guanine bases. Therefore, DNA is made up of 4 nucleotides: adenine, thymine, guanine, and cytosine. Meanwhile, RNA, adenine, thymine, adenine, uracil, guanine, and cytosine. So this will follow the rules of base pairing, in which that pyrimidine always base pair with the purine, in which adenine will bond together with the thymine with 2 hydrogen bonds. Meanwhile, guanine always bond with cytosine, forming 3 hydrogen bonds. And they have a sugar phosphate backbone, and DNA is arranged in a double helix structure, as you can see over here. Between adenine and thymine, they form 2 hydrogen bonds. Between guanine and cytosine, they form 3 hydrogen bonds. So how does DNA replicate? Remember, DNA replication is occur only during the interphase phase, which is specifically during the S phase, or known as DNA synthesis phase. So there are 3 proposed models or mechanism of replications of DNA. The first one is that conservative model, which discovered in the late of 1950s. So the conservative models of DNA replication showing that the 2 parental strands here, the dark blue color here, will reassociate, okay, after acting as a template for new strands. Okay, after they split together, they will reassociate again, and restoring its parental double helix here, as you can see over here. The original double helix will reassociate again after become template and forming the original double helix here. For the next one is semi-conservative model. For semi-conservative model, the two strands of parental DNA molecule here, they will separate, and each of it, the dark blue color over here, will become a template for the synthesis of a new and complementary strand, which is light blue in color here. Okay, and the third model is dispersive model of DNA replication, in which each of these strands of both daughter and molecules will contain a mixture of old and newly synthesized DNA. As you can see here, all of the replication result um, include the mixing in one strand, include the mixing of old and also the daughter strand. So therefore, the three types of models um, produced during the DNA replication are conservative model, semi-conservative, and also dispersive model. And Watson and Crick, semi-conservative model of replications predict that this one, okay, when a double helix that replicates, each of these daughter, okay, molecule will have an old strand here that being conserved from the parent molecule, and one is newly made strand that light blue in color. So competing model were conservative model, in which the two parent strand will rejoin, and the dispersive model, each strand that is mix of old and new strands over here. So for DNA replication, Watson and Crick model, which is suggests the semi-conservative model. Okay, and this suggestion has been supported by Matthew and also Franklin, as they did the experiment that support the predictions of the Watson and Crick. So semi-conservative, of course, from the parent molecule, they will separate during the separation process, and then the new strands here, they will become a template for a new strand. Okay. So steps in DNA replication. DNA replication is a process of copying a double-stranded DNA strand, just like a process of photocopy. So this process includes initiation phase, elongation, and also termination phase. So this is a summary of DNA replication. I will explain very simple about this one first before we go further into detailed explanation of this process. So what will happen? First, the helices here, the purple in color, they will unwind the parental double helix. Okay, they will unzip it, so the wound, and so the strand, these two strands will separated from each other. However, the strand is unstable. Then a single strand binding protein will stabilize each of these unwound parental DNA. Next is that primase will synthesize a short RNA primer here to start the synthesis process. And next is that the leading strand will continuously synthesize in the direction of 5' to 3' here by DNA polymerase. Meanwhile, for lagging strand at the upper part, at the lower part here, it will synthesize discontinuously. As you can see, there is a gap over here, and the primase, again, the primase will synthesize a short RNA primer, just like this one, which is later extended by DNA polymerase, same with this one, to form an Okazaki fragment. Okazaki fragment is referred to the short fragment of DNA. And then, after the RNA is replaced by the DNA, this one has been replaced by the DNA, okay, actually it's start from the RNA primer and being replaced by the DNA, by DNA polymerase. The DNA ligase here will join back all the Okazaki fragments to the growing strand. So let's look further on the further details on initiation step. So replications of DNA begins at special site that called as origin of replication, which the two DNA strands, that is the point where the DNA strands will be separated and opening up a replication bubble. Okay, I will show it to you later. An eukaryotic chromosome may have hundreds or even thousands origin of replication, meaning to say they will contain a lot of replication bubbles. So the replication will proceed in both directions from each of the origin until the entire molecule is copied. So as you can see here, they will form a lot of bubble here, okay? And then it will the origin of replication, so the DNA replication will continues at both sides of the replication bubble. At the end of each replication bubble is a replication fork. This one, the Y shape here, this one is known as a replication fork. Okay, so that is a place where the new DNA strand will elongating. And as I mentioned earlier, helicase are enzymes that will unwind or untwist the double helix at the replication forks. How? By breaking the hydrogen bond between the complementary bases, either between guanine and cytosine or adenine and thymine. And next, single-strand binding protein will bind and stabilize the single-stranded rDNA that have been unknown, and in which they help to hold the separated DNA strand in place so they will become stable. And next, the next protein is or enzyme is a topoisomerase enzyme. Topoisomerase enzyme helps to correct the unwinding ahead of replication forks by breaking, swaveling, and also rejoining the DNA strand, as you can see here. Okay, helicase will unwound the double strands, and then here single strand binding protein will stabilize the single strands of DNA that already being separated, and topoisomerase will help to correct the unwinding DNA strands. Next step in DNA replication is elongation phase. Elongation phase is referred to synthesizing a new DNA strand phase. So the primase enzyme is the one is very important, and also DNA polymerase. And these two are the important enzyme in elongation process, in which primase enzyme helps to make the RNA primer using RNA nucleotide to start the synthesizing process, and the primer will bind to the starting site of the replication. Okay, next is the DNA polymerase. So DNA polymerase, they require a RNA primer and DNA template strand to to synthesize the new DNA strands. What DNA polymerase did is that they will add a DNA nucleotide to the primer, and they will continuous adding DNA nucleotide complementary to the parental DNA strands. So the rate of elongation is about 500 nucleotides per second in bacteria and 50 per second in human cells. And the two strands of DNA in a double helix are antiparallel, So meaning to say they are oriented in opposite direction to each other. So if the DNA strand is 5 to 3 prime, the opposite strand is 3 to 5 prime. Okay, but the direction is always 5 to 3 prime. So the DNA polymerase can add nucleotide only to the free 3 end of the primer or the growing DNA strand. Thus, a new DNA strand can elongate only in the 5 to 3 prime direction. Okay, as you can see here, they start from 5 prime to 3 prime, 5 prime to 3 prime direction. The condensation process reaction will cause a phosphodiester linkage to form between one nucleotide to the next nucleotide. And DNA polymerase III enzyme catalyze the elongations of new strand at the location after the primer. So by continuously add nucleotide to the new complementary strand. So there are two types of strands that will be synthesized by DNA polymerase III, which are leading strands and lagging strands. Leading strands are the strands that growing towards the replication forks. Meanwhile, lagging strand, also known as Okazaki fragment, growing away or synthesized away from the replication forks. So all the primer are removed by DNA polymerase 1 later. Okay, so DNA polymerase 1 will replace the primer because it contains because it contains RNA nucleotide, and then they will replace the RNA nucleotides with DNA nucleotides. Okay, and next, DNA ligase. DNA ligase is act as a glue. Okay, in which they will join the Okazaki fragment or the lagging strand that being synthesized and also the leading strand. So as you can see here, for leading strand, they are synthesized towards here from 5 to 3 prime. They are synthesized toward the replication fork. Okay, they goes here toward the replication fork. Meanwhile, for the lagging strand, as you can see, this is actually here is a replication fork in which they will growing away from the replication fork, in which in lagging strands, then a polymerase III must work along with other template strand in the direction away from the replication fork. And the new complementary strand is known as the lagging strand, and this lagging strand also known as the Okazaki fragments. Okay, so what happened? The primer synthesize RNA primer, and then DNA polymerase III will replace the RNA nucleotide with DNA nucleotide and forming the first Okazaki fragment. DNA polymerase III will add DNA nucleotide over here after the primer to form a Okazaki fragment. Okay, and next, after reaching the next primer, over here, the DNA polymerase III will be detached, and then the fragment 2 here, okay, before the fragment 1, the fragment 2 is also being primed, and then a polymerase 2 will then a polymerase 3 will add the DNA nucleotide here and detach it when it reaches the fragment primer 1. So what will happen? Then a polymerase 1 will replace the RNA nucleotide at the primer, which is red in color, with DNA nucleotide. Okay, if there is a uracil, they will change it to thymine nucleotide. Okay, and then for DNA ligase, they will join the link between Okazaki fragments. Okay, So the comparison between leading and lagging strand, of course, as I mentioned earlier, leading synthesize toward the directions of replication fork, meanwhile lagging away from the replication fork. Leading synthesize continuously, meanwhile lagging synthesize discontinuously, forming the Okazaki fragment. And for leading strand, only one primer needed for the synthesis. Meanwhile, lagging strand, they need a lot of primer to start to synthesize the lagging strand. What similarity? Both use a lot of enzymes such as the primase to start the primer, the DNA ligase to join between two strands, helicase to unwound the double strand, DNA polymerase 1 to replace the RNA nucleotide with DNA nucleotide, and DNA polymerase 3 to synthesize to add the nucleotide to the after the RNA primer. And also both DNA polymerase 3 in leading and lagging strand can only add nucleotide to the 3' and produce leading and lagging strand from 5 to 3'. Last but not least is termination step of DNA replication. So termination step will make sure before termination, they will make sure that the nucleotide are paired up correctly. So the DNA polymerase will double check the sequence of the daughter strand or the new strand to make sure that the nucleotide are paired correctly. So DNA can be damaged by exposure to harmful chemical or physical agents such as the cigarette smoke, the X-ray, and therefore when exposed to these things they will can undergo spontaneous changes. Okay, so any mismatch that being detected will be repaired by the nuclease enzyme, in which the nuclease enzyme will cut the damaged DNA strand at 2 points and the damaged section will be removed. Next is that the DNA polymerase will repair by filling the missing nucleotide that being removed by the nuclease enzyme. Next is, of course, DNA ligase will come to seal the free end of the DNA to all DNA, making the strand complete again. As you can see over here, there is something that damaged here. So what will happen? Nuclease enzyme will come and cut the damaged part, and then DNA polymerase 3 will add the nucleotide here to replace the damaged one, and then A ligase will join between these two strands. So this is the steps of the Okazaki fragment. You have to remember later. So this is the simple steps in DNA replication. First one is involving helicase, unwound the parental double helix, then single strand binding protein, stabilize the unwound template strand, then the leading strand is synthesized continuously 5 to 3' direction or toward the replication fork by the DNA polymerase 3. Then primase will begin to synthesize the RNA primer for the fifth Okazaki fragment. Next is, of course, the DNA polymerase 3 is completing the synthesis of the fourth fragment. And then DNA polymerase 1, it functions to remove the primer, RNA primer, from 5' to 3' of the second fragment, replacing it with DNA nucleotide because we want to replicate nucleotide, not because we want to replicate DNA, not RNA. Okay, and last but not least, DNA ligase always will join the between two strands. Okay, so again, this one is the same.